Hello guys, today we'll talk about the digestive glands. Specifically, we'll talk about the salivary glands, the pancreas, the liver, the biliary tract, and the gallbladder. First up, we'll talk about the salivary glands and talk about the duct system in general, because throughout the whole glandular system, we're going to find a very common rationale and mechanism of how the ducts work. So let's talk shortly about the salivary glands. We have the major and the minor. There are three major glands, major salivary glands: the parotid, the submandibular, and the sublingual. And the minor, they are spread out throughout the whole oral mucosa. For example, they can be found on the palate or in the buccal mucosa. Now, let's take a look at where they are located. First, we have the parotid salivary gland, typically in front of the ear, the external ear, and we can actually see the parotid duct reaching out and opening in the inner part of the buccal mucosa. Then, of course, we have the sublingual gland located right here, exactly beneath the tongue, and lastly, the submandibular gland. There are, of course, major differences when it comes to the function. The structure is not so much different, but the function is, in fact. While the parotid gland is primarily a serous product, it produces 99.9% serous content. Of course, this means high amount of water along with a mixture of salivary enzymes. These can be antibacterial, they can be digestive. We'll see which ones they can actually produce. Then we have the submandibular gland, which is, of course, mostly serous and, of course, in a minor amount of mucus product. And lastly, we have the sublingual gland, which is mostly mucus and in a way lesser degree the serous one. So let's check out how these serous products come about, like how do they actually being produced. Well, we have the typical serous cells, the cuboidal cells. They are actually they resemble this pyramidal schematic, let's say. Of course, we have high amount of granular contents inside, of course basophilic, and this makes perfect sense because we have a cell that is highly productive and produces specifically enzymes. Of course, the enzymes are nothing more than polypeptides or peptides. So in this case, we can actually find extensive RER, extensive amount of course, ribosomes, and as a consequence, this high amount of ribosomes will have this basophilic and granular appearance. So this is how you can easily identify the mucous cell. Now, the serous cell. The mucous cell, on the other hand, has a very different appearance and morphology. We have this empty space-like empty-like spaces in the mucous glands, and we can see the nucleus, which is punched out on the periphery, which is, of course, there's a major contrast in the two images. And lastly, we have the in-between picture. This specifically the Serus Demiloon. Now, the term demilune means half moon, as the name suggests. Well, of course, here we find cells that are primarily mucous in the center, and surrounding encircling the periphery of the cell, we're going to find these serous cells hugging in a way, let's say, of course, these mucous cells. Now, typically, we're going to find one more cell in the acini, in the serous acini, or of course, the location of the productive cells, the myoepithelial cells. Now, these myoepithelial cells, for example, one is right here, a second one is right here. These are cells that practically extend and cover up the whole surface of the acinus, and upon stimulation they exert, of course, their contraction, and they push out the contents of this of the glandular contents. These cells are, as the name suggests, are epithelial cells with muscle-like contents. So let's talk a bit about the structures of the proportion of the acinus and the proportion of the ducts. Well, the question is, why do we even have ducts? I mean, it costs the human body literally hundreds of molecules of ATP, constant amount of mitotic regeneration and mitotic production. This, of course, is a costly event for the organism. But why do we need them, in fact? Why not simply put the acini closer to the locus of the location of the product where it wants to go and just end the story like this? Well, this is because of the fact that the ducts primarily have a modification function, meaning what? They regulate the amount of ions that are being lost on the product after, for example, the serous product. Well, because we have this excessive amount of production of ions from the serous cells or mucous cells, the function of the duct is to reabsorb ions, to reabsorb water in order to make the best possible outcome when it comes to economical terms. We want the human body to exert the least amount of work in order to have the maximal amount of output and the maximal output of work. So this is the function of the duct. So let's see actually what are the ducts that we'll discuss. We're going to talk about first off the intercalated duct. The intercalated duct is the first duct to be met exactly after the acinus. Exactly after that, we're going to find the striated duct, which is a bit bigger. Of course, this gradual processing, this gradual, let's say, classification and let's say this gradual increase in size and difference in the cells will actually also be visible in the macroscope. Meaning what? Well, typically the intercalated duct are going to have simple squamous or simple cuboidal. As we progress from the smaller glands, smaller ducts to the bigger ducts, we have this gradual increase in the size of the epithelium. For example, again, the intercalated is going to have typically simple squamous to simple cuboidal. Then the striated duct, and exactly next after that, is going to be a bit bigger. So it's going to be simple cuboidal to simple columnar. And then I'm going to find the excretory duct, which are the biggest ducts. And now here, in this case, we're going to find many exceptions. Typically, we're going to have, of course, stratified squamous non-keratinized epithelium, typically speaking. But of course, there are many exceptions in the human body, and you'll see them out throughout the whole process of studying. So this is a very nice schematic of the Serus Demiloon, exactly how it works. When I find again the cell and the Serus Demiloon hugging out the whole portion of the acinus. Now, this is a very nice schema. This is a nice graphic picture which actually shows the proportion of serous to serous mucus and only mucus when it comes to parotid, submandibular, and sublingual gland. This is exactly what I told you in the beginning. Parotid has 99.9% of serous. Mandibular has, of course, mostly serous and then a lesser amount of mucus. And lastly, the sublingual, mostly mucus, and then as it comes in a very short proportion of serous. Now, the graph is not exactly entirely true because occasionally we're going to find one or two very few cells of mucus cells in the parotid duct, but this is a part of the gland, but this is, of course, negligent. This is a very minor amount. This is a very nice, of course, graphic representation of the whole ductular system. As I said before, the intercalated typically simple squamous to cuboidal, the striated exactly the next one to simple cuboidal to columnar, and then as we grow bigger to go to bigger ducts, we're going to find, of course, pseudostratified, or in the main ducts, the ones that I told you before, the excretory ducts, are going to be in simple, typically stratified squamous non-keratinized epithelium. Now, there is a basic distinction we're going to talk about because all of the ducts, all of the glands specifically, all of the glands are, most of them are lobulated structures, which means there are specific lobules enclosed and encapsulated by, of course, dense connective tissue. Typically speaking, of course, not necessarily always. So we're going to talk about the intralobular ducts, the ones that can be found inside this let's say restricted space, and the interlobular, which is exactly between the different lobules we're going to find throughout the whole gland. So let's take, for example, the parotid gland. The parotid gland is of course an encapsulated organ, full, of course, covered by the dense connective, irregular connective tissue, and we can see these projections of this connective tissue called trabeculae, progressing and penetrating, of course, and forming the lobules, these organized enclosed spaces. So actually, let's see them macroscopically. This is exactly the picture of the parotid gland. Now, if we zoom in, we're going to find, of course, we can actually see, even by looking in a very minor degree of magnification, that most of these colors are basophilic, because all of these colors are, again, they're very high amount of serous cells. So in between them, we're going to find a bit more, let's say, acidophilic colors. This is, of course, these represent the ductular system, the duct system within the parotid duct, the parotid gland. So if we zoom in, we can actually see. So when we zoom in, we can actually see very nice and very clearly the very beautiful encircled, covered up serous acinus, and in the center, we're going to find the very opening of the intercalated duct. So how do we distinguish where is the intercalated duct, the interlobular, and so on and so on? Well, of course, all the areas that are enclosed inside the typical representation of the serous is the interlobular. This is a very cross section of the very center and the heart of this interlobular duct. Now, for example, here we can actually see the typical interlobular, which is we can actually see the cuboidal cells present being here. Now, the bigger these cells get, the bigger this ductular system goes, we'll actually be able to see the intercalated again here, and the striated ducts will be the columnar ones. For example, here we can actually see the intercalated ducts as the picture loads. So in this exact picture, we can actually see the continuation of the intercalated duct, starting from the center of the serous acinus and progressing throughout the whole ductular system. Now, as the size, of course, as the ducts progress, we're going to have bigger and bigger cells. typically columnar cells are going to be found in the striated ducts, and this is one example. A very nice encircled and very spherical representation of the ductal system, and here this is the perfect example of the nice myoepithelial cell that covers up and like practically encircles the whole duct, that allows for the contraction to take place and the content of the duct to progress throughout the duct. And lastly, we're going to find the interlobular ducts, which can be very easily found because they're actually very big in size. This is one right here, for example. This is a very nice picture of the typical interlobular duct. Now again, as I said before, the epithelium can be very, let's say, peculiar and not necessarily typical, but this is one example of how it can look like. Typically a striated structure present, of course, in interlobular between the lobules of the parotid gland. Now we can actually see a this part of the presentation is going to be very helpful for the ones that are still struggling to see the ducts. Now the serous acini, as I said before, in the center is the one that, this is the parts, of course, of the intercalated duct, and actually they're the continuation of the acinus, exactly the continuation of the products of the serous content in the duct. This is again the intercalated duct. Then we can find again the very nice columnar typical striated duct, and again surrounding them typically are going to be found the myoepithelial cells. Important also to notice that surrounding the ducts we can also find plasma cells. Now what do these plasma cells do? Again, we've discussed already the function of the plasma cells, which is the production of immunoglobulins. Now specifically, there's a very specific type of subcategory of antibody that's being produced and excreted throughout the whole ductular system, and as an excretory product, the IgA. Now the IgA can be found typically throughout the mucosal surfaces, for example in the oral mucosa, in the small intestine, and so on and so on. Now moving on, we can actually see the interlobular duct between the lobules of the parotid gland, or generally due to the generally present in the interlobular structures of the glands in general, and of course they have very atypical epithelial linings. Also, we want the submandibular gland. Now, as I said before, the submandibular gland typically produces mostly serous with a very minor amount of mucus. Now, the question is, how can you distinguish the submandibular from the sublingual, or the submandibular from the parotid duct? Again, the answer is proportion of serous cells to mucous cells. If you have a whole section filled with 99.9% serous cells, and all the rest of the features are, of course, point out to the serous content, then of course this is going to be your parotid duct, your parotid gland. Now, if you see a structure of a glandular structure, again, this is again the typical presentation of the acinus, with mostly serous, the whole picture being practically basophilic and minor areas being mucus, like empty-like cells, then of course this is going to be a submandibular. And lastly, if you find proportion, let's say 60% mucus to 40% serous, then this typically would be the sublingual gland. This is a very easy way to distinguish one from the other. Now, there's one unique structure here in the submandibular gland. Of course, this can be present mostly in the submandibular gland, and of course we talked about it, the Serus Demiloon, where it discussed what the function is and how it looks like. So this is a very nice mucus gland right here, and then circling right here is the Serus Demiloon, exactly right here. And of course, all the rest of the areas can typically point out to very easy to identify serous cells surrounding and serous acini. And of course, these are part of the ductular system. Now, judging from the size, this is a typical columnar cell, so I would have to say that this is the striated duct. This is how you should think. Based on the size of the epithelium, you should have the indication if it's the intercalated or striated duct, if it's inside the lobule. So this is the zooming and the zoom in, the magnification of the previous picture, exactly what we saw before. And again, we can find the typical mucus cell right here, encircled by the Serus Demiloon. Now, the Serus Demiloon is, as I said before, mostly a structure present in the submandibular. But this is not necessarily 100% true, because we can find very few, literally minuscule amounts of Serus Demiloons present in the sublingual gland. But typically speaking, you can find these structures in the submandibular gland. Now, this is again the sublingual gland. Of course, as I said before, mostly mucus. There we go. 60%, let's say, of the structure is mucus, and of course the rest is serous. Also, sometimes if you have a very nice section, you can actually see lingual, the parts of the tongue, the parts of the parakeratinized epithelium on the top. So this can be actually a very, very helpful hint for you if you're in doubting yourself, is it or is it not? Well, this is another element that you can check. Always, I told you before, I want you to, when you see a slide, I want you to think first, I want you to see all of the elements, all of the different tissues, and start to make up a whole logic rationale of the structure. I want you to think like a detective. You see, for example, the simple stratified, the stratified squamous non-keratinized, I want you to think this is a wet area, and so on and so on. For example, if you saw this section, you can actually see the pancreatic epithelium. This is again an area that undergoes friction, high friction, constant amount of friction. But because parakeratinized, there is of course a proportionally high amount of liquid in this area. You can actually see the highly glandular content. Of course, you can see the mucus and the serous, and now you know that this is a gland for sure. So the question is, which of them all? Because I can see the oral mucosa, the mucosa which can be found on the tongue, and now the question is, which of all? Now, of course, based on the proportion, you can judge if it's the parotid or the submandibular. This is how I want you to think when you see a slide. So let's go to the specific one, and we can actually see the very, very nice, clear picture of the mucous glands along with the ductular elements of the, this is the arteries possibly. This is possibly a capillary or an artery, you cannot tell, because we have no more indications whatsoever. So this can be either a very, very small duct, or this can be a very specific, like a small vessel. So again, this is, for example, the typical appearance, typical picture of the ductular system. This is again, this based on the appearance and based on the size, I could say this would be a striated duct, not necessarily, of course, being true. And this can be very, very easily distinguished. The cuboidal cells, this could be a very nice intercalated duct, and so on and so on. And of course, we can actually see the high amount of mucous glands spread out throughout the whole sublingual part. So let's move on to the pancreas. What is the function of the pancreas? Well, the pancreas is one of the most important digestive enzymes. Specifically, we can actually see that they produce a very wide range of digestive enzymes. Let's start with the proteases. The proteases, of course, are the enzymes that cleave proteins, as the name suggests. So this is a high amount of proteases can be found in the content of the exocrine pancreas. Now, why do I specify exocrine? Well, because the pancreas has dual function. One is the endocrine part, which produces the insulin and glucagon and so on and so on. We'll discuss this part in the glandular in another video. But now we're going to talk about the digestive part of the exocrine pancreatic secretion. That's why you focus, and I'm specifying the exocrine pancreatic secretions. So back to the content. Now, of course, the proteases are the ones that produce the enzymes that cleave the polypeptides to peptides, and the peptides to, of course, amino acids that can be easily and readily absorbed. Well, the alpha amylase. The alpha amylase is an enzyme that cleaves starch, specifically polymers of glucose, and they can, of course, cleave them in separate parts and absorb the monomers of the starch. And now, next up, we have the lipases. Of course, the lipases, as the name suggests, is the one that cleaves and is able to digest lipids, which can be cleaved into smaller, let's say, monomers, and then being absorbed not in the bloodstream, but of course in the lymphatic capillary throughout the whole lining of the small intestine. And of course, the nucleases. The nucleases are enzymes that cleave DNA and RNA. And now this is one of the most important things you will see, trypsinogen. Now, why is it so important? Well, because trypsinogen is the enzyme that starts and is responsible for the activation of most of the enzymes in the lining of the intestine. So one of the most important things we'll see in the pancreas is the trypsinogen. Now, what is the function of trypsinogen? Let's see actually. And as I said before, this is the most important part, the trypsinogen. Well, because trypsinogen is responsible for the activation of most, of course, digestive enzymes present in the small intestine. Now, the thing is that, of course, we already saw that the pancreas produces a very wide range of digestive enzymes. That ones that destroy proteins, one that destroy saccharides, and one that destroys lipids. Now, these enzymes are very, very potent. So if we have the activation of these enzymes outside the intestine, we're going to have literally autolysis, because the cell is nothing more than just a bunch of peptides, saccharides, and lipids. So it is very, very important. It's a highly important and highly it's imperative that these enzymes are activated only in the small intestine and not anywhere outside the digestive system, of course, outside the small intestine. So there are in fact 3 different ways to inhibit and to protect the ectopic activation of these enzymes. So first off, we have the trypsinogen, the grands of trypsinogen being produced in the pancreas, of course. They are activated by one very specific enzyme. We talked about it, enteropeptidase. Now, enteropeptidase is an enzyme located on the brush border of the small intestine. So this is one way that the human body controls the activation of trypsinogen only in the small mucosa. This is one way, because the enzyme that activates trypsinogen is present, of course, in the small intestine. Now, the second way that it protects this ectopic activation is the content of trypsin inhibitor being produced by the pancreas itself. So again, the serous cells, because again this is a gland, the serous cells of the pancreas produce along inside the same granule all of these enzymes, trypsinogen as well, and trypsin inhibitor. Now, and the last way that we control the activation, the ectopic activation of trypsinogen, is the low pH. Well, because we have the serous content, the serous cells of the pancreas produce also, not exactly the serous cells, the cells that are next to the serous acini, we'll discuss them which, are producing the bicarbonate ions. So of course they control the pH and they place the trypsinogen and the contents in a very, very specific pH in which, typically speaking, this trypsinogen cannot be activated. So we have 3 different ways: the location by enteropeptidase, the trypsin inhibitor, and the low pH along the content of the ducts of the pancreas. So this is a very, very nice graphic representation of how it looks like, of how the whole system of the acinus works along with the ducts. Now, again, these cells, of course, are acinar cells, of course, serous cells. Again, the typical characteristics: cuboidal, typically speaking, with, of course, with a specific taper, of the granular appearance, basophilic appearance, the same. And here we're going to find one unique feature, the centroacinar cells. Now, what does it mean? Of course, this is part of the intercalated duct. So why do we have a specific name? Well, because this is one of the conditions in which is, of course, it's a special circumstance. That's why there's a specific name always to it. So the centroacinar cells literally penetrate the whole acinus and ensure the very, very close, let's say, close control of the location of these products, of these granules. Of course, this is because of their high importance and high severity of wrong function. So these centroacinar cells that you can see in the picture right here, they literally penetrate the whole acinus and they perform one more function. They produce these bicarbonate ions, of course, again, to control the pH and to manage it to a pH that doesn't allow, of course, for the activation of trypsinogen inside the acinus. Ectopically, of course, ectopic from the small intestine. Now, the centroacinar cells are continuous with the intercalated duct, and again, we're going to find directly after this, we're going to find the intralobular duct. Now, it is very easy, of course, to identify the pancreas in the picture, in the slide. Well, this is because we have the endocrine content of the endocrine part of the pancreas. Now, macroscopically, in small magnification, we actually see that this resembles very, very much the parotid duct, because the parotid gland, because you can actually see high amount of serous, and again, a very nice capsule, the trabeculae. It's very, very similar. The difference is, of course, these sections right here, these lucent areas, where if we zoom in, we can actually see these are the pancreatic islets. This is the endocrine part, and this is how you can distinguish the, of course, the pancreas from the parotid gland. We'll see a picture specifically in just a bit. Before we go to the picture, I want to underline one thing, that we have, of course, an intercalated duct, but there is one thing missing. There is no striated ducts, meaning that we will not find intralobular ducts generally, of course, inside the lobular ducts, that are columnar and typically striated. So we're going to find only the intercalated duct. Then, of course, all the rest is exactly the same. From intercalated, except after the striated duct, we're going to find the interlobular, and the main pancreatic duct. Well, no difference whatsoever. This is a very nice picture that shows us exactly the difference of the appearance in the centroacinar cells. Well, because typically this should be the center of the acinus and should be empty. Here we see, of course, the presence of the centroacinar cell that is exactly within, of course, the acinus. So upon a cross section, we're going to find the nucleus of the centroacinar cell exactly in the center of the acinus. Of course, this is part of the intralobular ducts. This is, of course, the intercalated ducts. And this is, of course, the main excretory duct that we can actually very easily visualize in the macroscope. So let's check out the picture how it looks like in the macroscope.